We have previously designed a device to apply mechanical strain to adherent cells. In the current work, we have redesigned the geometry of the substratum and customized the setup for high-resolution imaging of these strained cells. The main advantage of this cell stretching system is that it provides a high-resolution subcellular imaging at 100x of the strained cells. Demonstrating the substratum fabrication will be Vicky, a research assistant from my lab.
Begin by designing a single-well polydimethylsiloxane mold by following along in the accompanying text protocol. Next, mix 20 parts of a polydimethylsiloxane base with 1 part curing agent in a disposable cup. Once mixed, place the polydimethylsiloxane mix in a vacuum degasser at -0.8 bar for 30 minutes to remove the bubbles from the mix. Then, pour the polydimethylsiloxane mix into the mold and the dish. Remove any additional bubbles at this stage by degassing it again at -0.8 bar vacuum for 30 minutes. When degassing is complete, bake the samples at 80 degrees Celsius on a leveling table for 2 hours. Next, carefully remove the samples from the oven and let them cool down to room temperature. Cut the edges with a blade and gently peel out the cured polydimethylsiloxane. On the 150 mm diameter polydimethylsiloxane, draw a 2 cm by 2 cm grid with a marker. Within each square, draw a 1 cm by 1 cm square